Celerion stands for swiftness and speed, and that is our defining principle to get your product onto the market faster. We are a phase 1 and phase 2 CRO with over 40 years of experience. And we are also a global company. Our headquarters is located in Lincoln, Nebraska, where we have a clinic as well as a bioanalytical lab. I'm based out of our clinic in Phoenix, Arizona, and then we have one other clinic in Belfast, Ireland. We also have a few offices in Europe, as well as a site here in Korea, in Seoul, and where we are able to work with some special populations. We also have a respiratory clinic located within our Belfast, Ireland site, where we have access to special populations such as patients with COPD, asthma, or cystic fibrosis. But today we're going to talk about non-alcoholic fatty liver disease, and that's where we have some of our patient groups located in the US. So to start, I'd like to just kind of overview what fatty liver is. And it's technically defined as having 5% or more fat within the liver. The increases of fat accumulation within the liver is associated with obesity, insulin resistance, as well as metabolic syndrome, and there are a few genetic factors as well. It's estimated that 25% of the general population has fatty liver. And that's defined as steatosis and having 5% or more fat accumulation in the liver by liver biopsy diagnosis. Now, 60% of people that have fatty liver or steatosis will progress to the more severe form of the disease, and that's non-alcoholic steatohepatitis. It's characterized not only by having fat in the liver, but also inflammation and apoptosis within the liver cells. Then 30 to 40% of patients can progress to developing fibrosis or tissue scarring. This can lead to NASH cirrhosis, resulting in need for liver transplant, end-stage liver failure, or even hepatocellular carcinoma. Here in Korea, the estimation of population with NAFLD is approximately 27%. And what's really interesting is that within that, 13% are not even obese. So there's some genetic factors at play for inducing fatty liver and NASH. Now, there are a number of different challenges facing the liver NAFLD NASH indication. And that includes the fact that there's no approved FDA drugs for this disease. Right now, it's currently being treated with diet and exercise. And patients that have type 2 diabetes may also be taking pioglitazone. Another major challenge facing this indication is the fact that there's no prognostic biomarkers. So we don't know who will end up just staying with having fatty liver for the rest of their lives versus those that will progress to the more severe form of the disease. And then finally, there's also a need for non-invasive biomarkers to actually track with some of the clinical outcomes, like cirrhosis progression, end-stage liver disease, and mortality. So this is a bit of a busy slide, but I'll walk you through it. It shows the pathogenesis of non-alcoholic fatty liver disease, as well as a number of different drugs that are at play and being developed to treat this disease. Now, I apologize if your drug is not up there, but it's a pretty crowded field, as you can see. So when thinking about NASH and its progression, there's really three main components. First is the metabolic component shown in green, and that's where adipose tissue triglycerides, free fatty acids from adipose tissue, can be stored and taken up in the liver where they accumulate. Now, the liver can also make its own fat through the process of de novo lipogenesis. And the bile acid pathway also plays a role. So you can see there are many different drugs acting along this metabolic pathway. Then there's the anti-inflammatory pathway, and that's highlighted here in black, where endoplasmic reticular stress, mitochondrial dysfunction, oxidative stress, pro-inflammatory cytokines all play a role in creating the inflammatory space within the liver. And finally, we have the fibrotic pathway, which is highlighted in blue. And these pro-inflammatory cytokines, as well as apoptosis, activate hepatic stellate cells to then produce collagen, which leads to fibrosis development. To actually identify patients with NASH, we need to do a liver biopsy. And we use the NAFLD activity score, or NAS for short. It is 3 components. First is the steatosis component, which is graded 1 through 3. Then there's ballooning injury, which represents apoptosis within the liver cells, as well as inflammation. So typically, a patient with a score of 4 or more indicates the presence of NASH. Fibrosis, on the other hand, is a separate stain, and this looks at the distribution and progression of fibrosis to cirrhosis. The more advanced form of this disease is really the bridging fibrosis, which is referred to F3, as well as cirrhosis, which is referred to as F4. Now, it takes approximately 7 years to progress from one stage to another, so this is a very slow chronic disease. And out of all of the histological features that we can measure for NASH, it's really the fibrosis that drives mortality. This is shown in a study here done by Angelo and colleagues, where he looked at patients with NASH or without NASH, and whether they had fibrosis present or no fibrosis present. And in the red and green line, those are the participants that actually had more fibrosis. They ended up having worse survival rates. So the liver biopsy itself has a number of challenges, and it's referred to as the gold standard or reference standard, but it is really a challenge because it only represents a very small portion of the liver, less than 0.02%. It also has low patient acceptance rates. So you might have a participant willing to undergo one liver biopsy, but they may not want to undergo the second biopsy after treatment because it is a painful procedure. It is quite invasive. There's also risk of complication and hospitalization with this procedure, and it's not suitable for short-term or serial measurements. However, the liver biopsy is the best tool that we have so far because it measures the activity of the disease. So it measures the steatosis, the inflammation, and the fibrosis. Right now, none of our non-invasive biomarkers can kind of do all of that. And as we saw, it also tracks with clinical outcomes. So what are the regulators thinking about how we can move forward in treating NASH? Well, the FDA released draft guidance in 2018 in December, and I have some highlights here. So for an early phase study, like a phase 2A study, the FDA is suggesting you could use either biopsy-proven NASH patients, or you could have patients screened with an inclusion criteria based on imaging modalities. For endpoints in these early clinical studies, they include AST changes, ALT changes, the changes in liver function test, as well as imaging modality and disease-specific biomarkers. So for example, say you have a drug that's acting along the pathway that affects apoptosis, then perhaps CK18 or caspase would be an appropriate biomarker for this endpoint. In phase 2b studies, this is where we really need to get into biopsy-proven NASH patients, and the biopsy results would be considered part of the endpoint. With phase 3 studies, now this requires, again, NASH liver biopsy to identify these participants, and the biopsy should be done within 6 months of actually initiating the clinical trial. Endpoints include resolution of NASH without worsening of steatosis, or improvement in fibrosis without worsening of NASH, or both, and then some of the hard endpoints showing clinical benefit. The European authorities also released some draft guidance as well. The EMA came out with a reflection paper in November of 2018, and they actually highlighted a number of similar points to the FDA guidance, but there are a couple key differences that I wanted to express. So first of all, the EMA is really considering having all patients diagnosed with a liver biopsy. For exploratory studies here, non-invasive imaging tools can be used. They define NASH as a NAS score of 5 or more, and they are looking for or recommend co-primary endpoints. So that would be the resolution of NASH and improvement in fibrosis stage. Recognizing that a drug that's really impacting, say, the metabolic pathway, the fibrotic pathway may not have effects on the metabolic area, here a 2-point improvement in fibrosis is recommended as an endpoint. Now, what both authorities agree on is that there is a very important need for non-invasive biomarkers, and specifically the need for diagnostic biomarkers, prognostic biomarkers, and those for patient selection. So now we'll get into what some of these non-invasive techniques are, and I want to start off by discussing the MRI PDFF. This is an MRI scan which measures the triglyceride protons relative to the water protons. And in this image here, you can see it's a cross-sectional slice of the abdomen showing the liver. The scale is from 0 to 50, and you have full coverage of the liver, which is much more beneficial than, say, just a biopsy, for example. And to compare these results with the liver biopsy, in the second graph, I have the histology steatosis grade on the bottom, 0 to 3, and the MRI PDFF results are highlighted. So you can see that there's really good separation among the 3 steatosis grades. This technique also has great performance, identifying patients within the various grades, and that's shown at the bottom with the area under the receiver operator curve. So it's been predicted that a 30% decrease in MRI PDFF would be associated with a 2-point change in the NAS score. And a number of sponsors actually did some preliminary exploratory analysis to see how their drug compared to this 30% change. They are all phase 2A studies looking at 12 to 24 weeks. The first is a thyroid hormone receptor binding agonist, FGF19 analog, acetyl-CoA carboxylase inhibitor, and toll-like receptor 4 antagonist. So after 12 to 24 weeks of treatment, you can see that almost 75 to 100% of patients were able to achieve that 30% reduction with MRI. The toll-like receptor drug, however, doesn't really work within that metabolic pathway, so there was really no change there in the results. So overall, the MRI PDFF is an excellent proof of concept tool, especially for drugs targeting that metabolic pathway. The next that I want to speak about is magnetic resonance imaging. Here, this is also an MRI type scan, and you have a patient lying in the MRI scanner, and the paddle is placed on top of their liver. The paddle is used to create a shear wave, and that propagates through the liver. So this top panel here shows the actual wave moving through the body and moving through the liver. And it gives us a value of shear stiffness, which is shown here at the bottom for the various fibrosis stages from F1 to F4. This measurement correlates very well with the liver fibrosis staining shown in this graph, and there's really good separation among the various fibrosis stages, as well as a very strong area under the receiver operator curve for identifying these patients. The number 19% reduction has been suggested as a value of change that would be significant and not due to chance alone. However, this number requires further validation with more studies. Now, there's one modality called the FibroScan, and that actually measures liver fat and liver stiffness at the same time. It uses a shear wave to measure liver stiffness and the cap to measure liver fat content. It's an ultrasound-like device with a probe that is just placed right up against the liver. It's very easy to use and can be used in the clinic right on site and gives you real-time data, which is a great advantage. Comparing the FibroScan to the liver biopsy results, you can see that there is a decent separation among the fibrosis stages, and it also changes with increasing steatosis grade as well. So the FibroScan, it correlates very well with liver biopsy, but it also correlates very well with an MRI scan results for measuring fat content. And that's shown in this graph here, that the area under receiver operator curve is quite high when you're comparing the FibroScan results with an MRI scanner. So considering a clinical study where you want to enroll patients that have have an inclusion criteria based on MRI PDFF, then pre-screening with the FibroScan is actually one way to help expedite your trial, because you already have these patients pre-selected before they go into the liver scan, which is a more expensive type of analysis. So you want to be sure that the patients that you are screening for your clinical studies are going to be within your IE criteria. And part of what we're doing at Celerion is we're building a database where we are actually proactively pre-screening participants with our FibroScan. They also get their clinical chemistry labs done, and we measure some biomarkers such as CK18, some of the pro-inflammatory biomarkers as well. And this is great because, as I mentioned, it really reduces screen failure rates before moving into some of these more expensive type inclusion criteria aspects, as well as expedite enrollment. So, for example, if you using the FibroScan, you could probably pull your study off with 1 to 2 sites, whereas if you're looking for biopsy-proven NASH patients, then you need a multi-site type plan. The last imaging modality I want to mention is the liver multi-scan. And this is also a MRI type scan. It measures the T1, which is a component of the MRI scanner. And this needs to be corrected for iron. So it's a measure of water corrected for iron, which gives us information about both fibrosis and inflammation. This modality has a fairly good area under the receiver operator curve for identifying patients with NASH, so not just steatosis, but also the disease as well. More validation is required as only a handful of studies have used this during a clinical trial. And then there are some soluble biomarkers that reflect the various pathophysiology of the disease. So biomarkers for oxidative stress or liver fibrosis, inflammation, and apoptosis can also help in your clinical trial to judge efficacy. Two that I want to highlight is the ProC3, which is an excellent biomarker for fibrosis, and CK18, which is a very good biomarker for apoptosis or for NASH. And the Korean non-alcoholic fatty liver study group actually proposed a few outlines for study design. So with early clinical phase trials, no histological evaluation is needed, but inclusion criteria could be based on the MRI PDFF, liver enzymes, and MRE, for example. Getting into some of these later phase studies, that's when, again, you really need to use the liver biopsy, but this can be combined with some of the non-invasive modalities as well, which is handy so you can track your drug along the way and see how efficacious it is. So to summarize, the liver biopsy is still required for inclusion and exclusion criteria. But when conducting early clinical trials, phase 1 and phase 2A type trials, here the non-invasive biomarkers can really be used. The MRI PDFF correlates very well with the steatosis grading by liver biopsy, as does the MRE. However, further validation is required to get an idea as to what that % change should be that we're looking for. And there's a number of different projects underway with biomarker consortiums who are hoping to address that. The FibroScan, as I mentioned, is an excellent pre-screening tool and can also be used as an exploratory analysis in your clinical trial. So I would like to thank you for your attention and happy to answer any questions you may have.
Any questions? Any comments? So do you guys have any questions? Yeah, we have a question over there. We're going to deliver the microphone up back there. Yeah.
Yes, really, really nice presentation. Thanks very much. The disagreement between EMA and FDA regarding biopsies, what do you think the root cause of that is?
That's a great question. It's hard to understand sometimes what the regulators are thinking, but I believe what they want to do is make sure that the right participant is being treated. There was a study done by the GenFit group. Their drug is elafibranor. They actually had allowed patients with F1 through F3 in their study, and there was no real change there with the earlier fibrosis group participants. So I think the EMA is being very conservative and in trying to hope that the patients that will be taking this drug will receive some kind of benefit.
Thank you. Very good question to answer. Any other comments? Any question? Okay.
Hi, my name is Kaza Oyagi, working for Celerion. If you like a copy, PDF copy of her presentation, please stop by before you go out and give me your business card, and I will mail it to you today. Thank you.
Okay, thank you very much.